Welcome to STEMpaths, where we unpack papers, we have students look through them, and we unpack the authors behind them. Today, our guest hosts, we have Jailyn and we have Chloe, and they're going to introduce our guest author today, Dr. Connie Mulligan of University of Florida, on her paper, Low Birth Weight is Associated with Epigenetic Age Acceleration in the First 3 Years of Life, from the journal Evolution, Medicine, and Public Health from 2023. If you guys want to take it away, go for it.
Okay, my name's Jailyn. I'm a psychology major, and I think my career paths is centered around healthcare, like we were discussing at the beginning.
My name's Chloe Aguilar. My major's animal science, and so I just think that all this information is really cool, especially with epigenetics.
Yes.
So you guys want to give a quick summary of the paper?
Yes. So I don't know if you want to go.
Okay, yeah, I'll do my brief summary real fast. So this study basically just shows that infants born with low birth weight experience accelerated biological aging during early age childhood, suggesting that early life adversity leaves a measurable epigenetic imprint that may shape long-term health and disease risk.
And what I got from the paper was just how the researchers wanted to see if low birth weight, which is often a sign of early life adversity, could actually change how fast a child's biological clock ticks through changes in DNA methylation, which is crazy. And basically, you guys were trying to find out if babies who are born smaller start aging faster on an epigenetic level, even as toddlers. And the study was conducted with 67 mothers and using the epigenetic Hovart's epigenetic clock. And you guys found that babies born with low birth weight were more likely to show signs of faster biological aging by age 3. So I don't know if we touched on your paper very well. Did we miss it?
No, you got all the major points. That's exactly right. So I can expand on it a bit more.
Yes.
But first, I'd like to ask you guys a question. So what was it about the paper that kind of intrigued you? It sounded like maybe this increased aging thing, right?
Yeah, I think I really was drawn to epigenetics, like, while taking this class. I liked, just how, like, your sequences can be, biologically, like, the same, but the gene expression can just come out differently. So just the fact that aging can be different and like, express later on different, like, diseases down the line, and I don't know, I just thought it was interesting.
Yeah, I think it's kind of crazy that something that you, like an epigenetic imprint, can affect you positively or negatively so far down the line of your life. So, like, if you get an epigenetic imprint when you're 2 years old, it can affect you whenever you're 54 years old. That's my understanding of it, and I think that's pretty crazy.
Yeah, that's an excellent understanding of it. So what actually got me interested in this whole line of research was the impact of stress and trauma on our health. So we all know that stress and trauma, at least too much of it, negatively impacts our physical and mental health, and possibly even decades into the future, and possibly even into the next generation, which is kind of wild to think about, that how can something that happened, far in the past affect our health so far into the future and possibly into our kids and grandkids. Mm-hmm. And so I was, and so from a kind of simplistic perspective, like if a woman, when she's pregnant, doesn't get enough to eat, you can understand that would negatively affect her baby. Baby's probably going to be born low birth weight, and we know that low birth weight is an indicator of poor health throughout the lifetime. But as a geneticist, I was really interested in, but how does this happen? How is this information about, and so the example I gave about a pregnant woman who doesn't have enough to eat, that's kind of easier to understand, that she literally was starving, so her baby was starving. But if it's more like psychosocial stress or trauma, then it's a little harder to understand what the molecular mechanism is that takes that information about that stress and trauma and somehow preserves it so that it can have an effect for decades. And so as a geneticist, I'm thinking about, well, what could it be? It can't be the genome, because the genome's already set, right? So that's why epigenetics came to mind. So epigenetics is epigenetic modifications or alterations that are made to the genome that don't affect the DNA sequence, but do affect the expression of genes. And so we studied DNA methylation, which is literally just putting a methyl group on, most often it's cytosines that are followed by a guanosine. And that methyl group then is known to affect the expression of genes. If you've got a lot of methylated cytosines in the promoter upstream of a gene, those methyl groups literally get in the way of all of the transcription machinery and impede that DNA then from being transcribed into RNA and then translated into proteins. But even DNA methylation in areas outside of the promoter, anywhere in the genome, is thought to affect gene expression, though it's not quite as simple a relationship as more methylation leading to less gene expression. So anyway, to me, DNA methylation just looked like a perfect candidate to store this information about stress and trauma in a way that could possibly persist throughout our whole lives and possibly even be transferred to future generations. And so I really got interested in this idea of DNA methylation being this mechanism that sort of mediates the impact of stress and trauma on health about 15 years ago. And I started working in the Democratic Republic of Congo. I actually had a grad student who had started working on this. And I will admit that she was a first-year cultural anthro grad student, so really no background in genetics. And she was taking a course I taught that was required for first-year grad students, and she mentioned epigenetics. And I was a little judgmental, I admit, and I thought, you're a cultural anthropologist. What do you know about epigenetics? And it turned out she knew a lot. She knew a lot, and she knew that her project was perfectly, positioned to study this effect of, in the shared environment of a 9-month pregnancy, exposure to stress and trauma, and then also, the developing fetus having its DNA methylation marks laid down for the first time. So we started collaborating, and I've been working on the project for 15 years. And so the paper you guys found is a more recent paper by another grad student of mine, Edward Quinn, who is amazing. And so what he found, I'll tell you. So we were looking at this effect of stress on health and looking to see if DNA methylation sort of mediated that effect. Well, we didn't find any association between the stress that our mothers from the DRC had experienced and DNA methylation. But then we also tested for an association between DNA methylation and then our outcome variable, which was the baby's birth weight. And then we did find that association, like you said, it was the babies who were born low birth weight, then over the first 3 years of life, experienced accelerated epigenetic aging. And so the way we calculate epigenetic age, we look at how it correlates with chronological age. And there's a really good correlation, which is not surprising. But then we look for any accelerated or decelerated aging, that's sort of beyond that correlation with chronological age. And when we looked at the babies who were born low birth weight, we found that they did have a statistically significant, acceleration of epigenetic aging. And what was interesting was that we only saw it in the trajectory. So we sampled the baby's blood when they were born. Then we got them at 6 months, 1 year, 2 years, 3 years, and 4 years. And if we had looked at any one of those time points, we wouldn't have seen any epigenetic age acceleration. It was only by having all those time points and looking at the trajectory that we had enough power, statistical power, to see the effect of aging. And so we then hypothesized that, from this very early age, literally from the time they're born, they seem to be experiencing accelerated biological aging, which we speculate is not a good thing. In babies, maybe aging is different. But in general, we sort of think of accelerated aging as being a bad thing. But I will say it's still an open question. Could that accelerated aging in some way be adaptive? And I think we mentioned that just a little bit, that we're not exactly sure what accelerated aging means, but that it might be a way in which birth weight can impact health decades into the future if from birth this accelerated aging imprint is made.
Okay. Thank you so much for sharing.
Yeah, that was a lot of information.
Like I feel like I just read the paper all over again. I was really surprised when you were talking about your emphasis on this, like the stress and trauma. Because I'm a psychology major, so it's like you're speaking my language now. So I'm glad that's like one of like something that inspired you in conducting this research. And I don't know if you had any other thoughts about why you wanted to focus on the stress and trauma in conducting this. Like it could have been any other factors with regards to aging, but you chose stress and trauma and nutrition as well to focus on as your factors.
Yeah, I mean, I guess I look at stress because, like everybody's stressed these days, right? I mean, we always talk about, oh my God, I'm so stressed. And, we generally think of it as being a bad thing. And certainly too much stress is a bad thing. But if we didn't have any stress, but there is some kind of good stress, good stress that gets us out of bed in the morning, makes us want to go and do stuff, accomplish things, so it's sort of, you got to have the right balance of good stress and bad stress. Mm-hmm. But that said, there's certainly, this idea that everybody's stressed. Oh my God, we're stressed. Why are we stressed? And what's the effect? It's thought to have a negative effect. And I guess also I had 3 teenagers at home when I was starting this research. So I also think about, I'm stressed, they're stressed, we're stressed.
To study this.
And, to try and study it, to try and better understand it, and to maybe, find a way to mitigate the bad effects of stress. So, one project I never got to do with our moms in the DRC was, so I'd like to think of interventions, ways that we can, weaken or mitigate the bad effects of stress on health. And, so you think of good interventions like exercise or meditation or spending time in the outdoors. We think of the healthy things we try to do. And there are some people who've studied those sorts of phenomenon to see if they then get epigenetic age deceleration. Like, does it make us age more slowly if we do yoga every day or something like this? That's really hard to study because it's such a long-term thing, and it's just really hard, So even if it does have an effect, we're going to need a lot of samples. We're going to need a really controlled sort of experiment where everybody's doing their yoga or their exercise the same time, same, number of minutes a day. It's just from an experimental study perspective, it's hard to do that. Yeah. But I was thinking, in our project in the DRC, what might be interventions we could try, and one that came to mind would be breastfeeding. So we know that breastfeeding is really great for the baby. It's the best nutrition that the baby can get, for like the first months or first year of life. And, it would be an easy thing to add to my study. I haven't gotten funding to do that, is sort of the short answer to why I haven't done it. But, I'd love to look at something like that to see if that then seems to weaken the effect of either stress and trauma or the epigenetic age acceleration. So I guess, ultimately, I'd like to think that if we better understand how stress impacts health, we might be able to identify interventions or probably more near term, identify individuals who show epigenetic marks of stress and trauma, and therefore might be, more susceptible to bad effects of that, and then might be someone we'd want to direct into sort of different intervention programs.
Yeah, that's awesome. Thank you for sharing. I wanted to invite us to go back into the paper and just kind of, I got a little bit ahead of myself on asking those questions about, but I think it was really good that I asked about your motivations as well. But I didn't know.
Yeah, I kind of have a question about your study design. I was just kind of wondering why you did choose to do mother-infant pairs in the DRC. Maybe if you could elaborate more on that, because I know they're more likely to, I wouldn't say struggle, but more likely that their infants are more like low birth weight or bad health. Mm-hmm. But it's kind of, I guess maybe it is just like your population size would be too big if you did choose to like venture out to even like higher income people who maybe are still struggling with the same things, and maybe it's not, the income, but maybe it's like body dysmorphia and, more physiological, types of mindset. So I kind of just want to understand why you did just choose to do the DRC.
Okay, well, the short answer to that is that it was my grad student who first brought up the idea, and she already had a project going there. So it was just easy to add in, an epigenetic component. But more from big picture of, how to design a research project. Again, it has to do with what I was talking about before in terms of how difficult it is to study the effects of something like yoga or meditation that are just kind of, it's almost like trying to catch smoke, right? In terms of really identifying an effect of something that's probably going to be pretty mild. And so I reasoned that we needed a pretty strong stressor in order to have a chance of identifying something. And unfortunately, women in the DRC are really stressed. So there's political violence, there's sexual violence, there's war violence, there's grinding poverty, there's a huge infectious disease load. So from that perspective, I thought if we could study a really extreme stressor and find some key DNA methylation marks or, DNA methylation phenotype like the epigenetic age acceleration, then maybe we could study milder forms of stress. And given, being able to focus in on the results we found from the study with our more extreme forms of stress. So that was kind of the logic behind it. We haven't actually gotten to the point that I just described. We haven't identified key DNA methylation sites yet. And partly that's because we really don't know how methylation works. We don't know if a region of the genome needs sort of an average level of methylation. And it doesn't really matter which sites are methylated so long as you have that average level of methylation. Or are there key sites that have to be methylated? So we understand DNA methylation and the promoters of genes pretty well, but that's just, genes only make up 1 to 2% of the human genome and promoters even less. So that means, over 99% of the genome, we don't understand how DNA methylation influences gene expression. So I haven't identified those key sites, that we can then go and test against milder stressors. But that was basically the reason why we worked in the DRC. And I've got to say, once I started working in the DRC, we just have a wonderful research team, and our moms are great, and they're so committed to the project. So it just turned out to be a wonderful place to do the research.
That's awesome. I wanted to ask, I know that there's like some limitations in the research as far as like definite answers as to what is like the cause and everything, but I do want to ask, a little bit more your insight on accelerated aging. Just like, what do you think it could cause? And in regards to like the stress and nutrition levels.
Right. Yeah. So, it's a hard thing. So our study was the first to look at those really early years of life and get multiple time points. So we identified, we filled a gap in knowledge, by looking at those really early time points. But it'd be great if we could watch those kids through the next 5, 10, 20, 30 years to see if that age acceleration persists. Or maybe they reach an age where it just kind of disappears.
Yeah,
and then it would say, so then that would sort of say, well, yeah, the bad effects of, low birth weight are present in those early years and could certainly have an effect on health, but that, maybe that epigenetic age acceleration doesn't persist through life. But we don't know that, nobody's done a decades-long longitudinal project like that. Now, that's another project I'd like to do if we got funding, go back and, get some of our moms and kids, now that we started this project in 2010. So, we've got teenagers there. It'd be great to look at them. And that's another good thing about having worked in the DRC, that our research team is still in contact with a lot of the moms. So we still could, contact them and possibly, get more samples if I got more funding. So we don't really know that. And then there's also a line of thought that sometimes rapid aging early in development could be seen to be adaptive. It's like if you're born into a really stressful environment, maybe it's best if you just develop and mature as quickly as possible so you can fend for yourself. So that's what I was alluding to earlier, that we're not positive that epigenetic age acceleration in ages from birth to 3 years old is necessarily a bad thing. It could be adaptive, but it's very hard to test that.
Okay. Well, thank you for giving me clarity on where the research is at.
Yes, no problem.
Did you have anything?
Do you guys have any other questions?
I think we have a few more.
I would just ask, how these findings help us understand childhood development in these low-resource environments, especially since, they are so low-resourceful, they don't. Obviously, it is kind of like a blessing for y'all to come in and do this just to see, like you just said, if it could help them, age faster and, fend for themselves. In most cases, that is a good thing for these kinds of environments, but I'm just kind of wondering, does it help us understand the development in these low resource environments?
Yeah, so I guess I wouldn't necessarily say that, like I said, we chose to work in the DRC because we wanted this really extreme stress phenotype. And in terms of working in a, low resource, high violence, high poverty, environment, the one thing that sort of struck me that I've actually got some students starting on a project now. So we know that stress affects health. We know it affects the health of a pregnant woman and the health of her baby. And we've certainly found that in our study. But what is almost more striking is actually how healthy the babies are. So despite, war violence, sexual violence, political violence, poverty, infectious disease, all of this, the babies are pretty good. So even that low birth weight, it wasn't stunning low birth weight. And even the association with epigenetic age acceleration, it was only a p-value of 0.01. So it wasn't knock you over the head. Oh my God, these babies are barely surviving. I mean, that's not the case at all. And what's interesting is that whenever I go back to the DRC, our research team always invites in some moms and their babies for me to visit so that I can, I want to see them. They want to see me. I want to see how their babies are doing and stuff. And in general, the babies are doing great, despite where they're living. So what has more struck me recently is, how are they doing so well? As a scientist, what am I missing here? That I've got all these data on the horrible things that are happening to the moms and their babies, and yet the babies look pretty good.
Mm-hmm.
So I must be missing something, that I'm not getting this linear relationship between all the horrible things happening and, poor health. Mm-hmm. And so what I'm looking at now is getting better data on resilience and coping resources and, positive things in the moms and babies' lives that help them overcome all of the adversity. So again, like as a scientist, I think about, we have hundreds of questions on the negative influences in our moms and babies' lives, and literally about 10 questions on positive things. Mm-hmm. So that's a huge oversight on my part. And so we're working on going back and asking them questions about the good things in their lives that help them persevere and, presumably help them survive and even thrive. So, I almost wonder if, as opposed to me giving something to them, if they can give something to me in terms of better understanding what sorts of positive influences, positive factors can help overcome negative factors in our lives.
That's awesome. Yeah, you answered a lot of my questions that I was going to follow up with.
Oops, you guys just froze.
Mine too.
Oh, you just
Can you
froze.
hear us?
Did I freeze too?
No, you didn't.
You slowed down briefly, but you're not frozen.
Okay.
Here, let me help them pivot a little bit here. You're so enjoyable to talk with. I'm wondering what some of your ways of communicating research findings are when you get a good run on research finding. What it, through either professional settings or even lay settings, what have been some of your more enjoyable ways to communicate research findings?
Wow. I think I'm pretty standard, writing papers and stuff. I do like giving interviews and stuff like this. We had a paper that came out last spring about epigenetic signatures of violence that persisted, that appeared across 3 generations. And what was kind of interesting is that, I'm now 40 years into my career, over 100 articles I've written, and that was the most impactful. That one got the most attention from the press. I got requests from interviews from around the world, and it was kind of startling, but also kind of fun to, hear from people around the world who were asking, to talk to me about that paper. Because I do think that that's the only, like I say, I don't do that much outreach. I don't have a podcast. I don't do social media stuff like that. So it was great to know that somehow other people were, getting my paper and getting the word spread through social media. So I enjoyed doing those sorts of interviews. And I guess just in teaching, you know.
You hit on something interesting to me also with the idea of this paper kind of took surprise at how it kind of grew wings and flew. But of all the 100 papers you've written, what one did you think might have had the most impact compared to the one that actually did?
Oh, that's hard to say which one.
I'm sure it's a little challenging. I'm sorry.
Impact. I mean, so the paper I was probably most excited about was the very first paper I wrote on this project in the DRC, where we first found this, evidence for a DNA methylation signature that mediated between maternal stress and infant birth weight. And it's been really highly cited, but that's really just in the scientific community. It didn't really make it out into the general public. And, I don't think it was any more dense or difficult to understand than the one this spring that made it out. So I guess I don't understand really very well what captures, the eye of people out in the general public so well. But anyway, yeah.
Right, exactly. What is it that catches the eye of those people who take our work and are able to communicate it more to a broader audience? What do they see in our work that we're not able to communicate that same way almost?
Exactly. They had a workshop. Our College of Journalism and Communications had a workshop for faculty to learn how to better, communicate our research to the general public. And so they did our makeup and had us, with lights, like we're being interviewed on TV, this kind of stuff. And so they had me talk about my research, and I explained it, very similarly to the way I explained it to you guys. And then they explained it back to me, like how they would present it. And I'm like, oh, wow, that sounds so cool. I was just amazed at how they took what I did and made it sound so much more interesting than I had made it sound.
Yeah.
So, and I guess that's their job, right?
Absolutely.
So I do like being interviewed, and seeing how they, how they make it much more accessible, much more exciting to, someone who isn't a genetics geek.
Yeah, that's so true.
I kind of had a question. I know you just said you get asked for interviews all over the world. What's your thought process when you saw Dr. Larson's email about a little university, little two college students wanting to interview you? I mean, you could have easily ghosted us like those two other authors did.
Yeah, you're on a boat right now. I wouldn't be surprised.
So I'm just kind of wondering, is it like, oh my gosh, this is so exciting? I just want to know your thought process, kind of.
Yeah, well, I have to say your professor wrote a good email, because I read it and I thought, oh, that sounds really cool, having students in the class do a podcast and interview faculty. And I thought it sounded like a really great forum, because, to me, it was more interesting than a student just saying, I have to interview a professor and write, a paper for my class, because I get a lot of that. But this seemed much more interesting, making it a podcast. And so I think that really caught my eye, and I'm like, yeah, I'll do that.
Yeah, I'm pretty grateful for this experience too. I thought it was going to be more rigid and that I wasn't going to feel more natural talking to you. But I think even just your personality, it's very easy to just ask questions.
Yeah, you're very charismatic, and the fact that you're on a boat in the middle of the Atlantic is so cool.
That's kind of cool, huh?
Yeah. I wanted to ask you something. It's a little off topic, but it's been on my mind. I don't know if you have any pets, Do you have any pets?
Oh yeah, I have a dog, Max. You want to hear the story about how I have a dog?
Yeah.
Yes.
So I have three kids. They're all grown and out of the house. But my oldest, my daughter, was allergic to dogs, and so, or well, allergic to all pets. So we never really talked about having a pet. And then when she started in college, I guess maybe in the back of my head somewhere. But then my two boys, my younger kids, both sons, played soccer, and I was at one of their soccer games one day, and it was raining, and his best friend had just gotten a little dog. And even though it was raining and he was all bedraggled and, soaking wet, I thought he was just the cutest thing ever. And I thought, that's a cute dog. We could get a dog. We should get a dog. And so over the space of the weekend, my husband and I had always been, we don't need pets. We got three kids. We need pets like we need a hole in the head. So over the course of the weekend, I'm like, we could get a dog. We should get a dog. Let's go get this dog. And my husband's kind of like, what's got into you? And so we got Max. And I always say Max was my best poorly thought out decision ever.
I feel you on that one. I have a dog named Max and his brother Artie, and it was very poor decision, especially as a college student with no money. I don't know why I did it, but it was a good decision. But I wanted to ask you about if you had pets, because with the infants and birth weight, in regards to epigenetics in this study, if I've always been so curious about how dogs and epigenetics, how the runt, for example, and even just accelerated age, I feel like they can all kind of correlate, how I think runts with not having a lot of nutrition, how their gene expression is very different. And I have a dog like that where I have his brother Max and his brother Artie, and they have two different gene expressions. His brother Max, he's perfect, he's nice and he has a lot of muscle, but Artie is also, he's super skinny. And when I was reading this article, I was like, wow, even with pets too, I just thought it was cool. Did you have any thoughts on that with pets and epigenetics?
Yeah, I mean, I think so long as we're talking animals that are still sort of evolutionarily close to us, the sort of results we find in humans could also, be true for them. So yeah, the different phenotypes in, dogs from the same litter could be some influenced by nutrition, which I think is how differences are typically explained for, in the same litter. But maybe we're wrong on that. Maybe it is other things, more like changes in gene expression, some of which might be regulated by epigenetics. I don't know. It's, so I think there's a lot there. It's really hard to study. First of all, it's expensive. They're expensive data to generate. So you've got to have NSF or NIH funding to have enough money to do it. And then the phenotype, you just really got to have a strong and then sort of sharply circumcised phenotype.
Yeah.
And I think that's what makes studies hardest to design.
Yeah, I heard you mention just
circumscribed. I knew circumcision. Yeah. Anyway, go ahead.
We understood what you meant.
Yeah, we did. Yeah, was it hard for you to narrow down on birth weight or on low birth weight as well, being one of the motivators?
Well, I mean, so in terms of health outcomes, we focused on birth weight because we know that birth weight, it does correlate with lots of, later life health diseases and conditions. And birth weight's easy to measure. Is the bottom line. We actually bought a second scale for the nurses at the hospital we worked at in the DRC so that we could get 2 measures of birth weight for each of our babies so that we could make, and then, we average it and make sure there's really good correlation between the two, because we need to have really good data in order to see anything, right? So we've been focused on birth weight for a long time. And so then it was kind of obvious to look at low birth weight, to be quite honest.
Yeah.
Once we were analyzing all of our data.
So we know you're on a boat, and we know we're running out of time here. So we just got our 3 last wrap-up questions that we ask all our guests for some, continuality between podcasts. So I'll let them ask those last 3 questions.
Okay. So what is one piece of advice you'd give your undergrad self?
Okay, so what I tell undergrads and what I did as an undergrad, but to be quite honest, I probably would have freaked out if my kids had done it as an undergrad, is that I definitely followed my gut on a lot of things. If I look back on decisions I made, like why did I interview for the honors biology program? It was life-changing, but it literally was such a casual decision. I'm walking down the hall, I see a little paper up on the wall saying interviews for honors biology program. I'm like, I'm not busy. I can go do that. I mean, it was, again, this incredibly poorly thought out decision, but it was life changing because we did research. It's what got me to go to grad school. It's, so I try to think back and, I wasn't really a frivolous, unthinking sort of person. But I think I was very open to possibilities, open to new things, open to adventure. So I think I would say that, be open to adventure, because there's so much pressure on you guys from middle school or earlier. Pick a career. What are you going to do with your life? Take all kinds of AP classes in high school, graduate college as fast as that's kind of all nonsense. Because you've got to be open to different possibilities. And I think all of the pressures on you these days make it very hard for you to be open to possibilities, and particularly open to the idea if someone says, well, what are you doing that for? And if you don't have a really good idea, they're going to make you feel really bad for doing that. And if you say, well, it just sounds good. Yeah, that's not going to... Yeah. Anyway, I would encourage you to push back on that and say, it feels like the right decision and I'm going to see it through.
Yeah. Sort of like a leap of faith, like just let life happen. I feel whatever you just said because I'm about to graduate next week. So everything you said, I'm like taking in. But one of
Malcolm Gladwell wrote a really... Sorry, just one more thing.
No, it's okay.
Malcolm Gladwell wrote a really interesting book called Blink, and it's about making decisions, making big, important decisions. And he basically endorses this idea of feeling your gut, following your gut. And he's very against making pros and cons lists. And I'm kind of the same, because sometimes you do a pros and cons list and you get to the bottom and you see what you're supposed to do and you get this sort of sinking feeling in your stomach. And you're like, oh, well, I guess I'm going to marry that guy, because that's what my list says. So anyhow, I never do pros and cons lists. I try to learn how to listen to my gut.
I'll try to keep that in mind.
That's noted for sure.
Yes. So if you weren't doing this and your research, what would you be doing for your career choice?
Oh, wow. Well, I will say when I went on my first professional conference, I was so turned off by having to sit in the audience and listening to all these talks that I thought were quite honestly quite boring. And I made a list of alternate careers, and it was open a flower shop, and, I don't know, all kinds of things that were not a scientist at all. And then I realized that you're not actually supposed to go and listen to all the talks. You're supposed to network and talk to people and, go to the stuff that interests you.
Oh.
But I was not always committed to being a scientist. I will say now that I'm at the end of my career and thinking about what to do and if I want to do something next, I read a really fascinating book about a woman who is a hospice nurse and how she gets so much, enjoyment and validation from helping people who are dying, spend their last weeks. And she also talked about amazing experiences she's had with people, who claim to be able to see on the other side, see people who have died and who they're now feeling closer to those people than the live people in the room. And so I thought, wow, if I've got time for another career, that might be a fun one.
Yeah, that does sound really deep.
Yeah, it does.
Yeah. Yeah.
Well, with your career choice,
We just have one more question, and it's what's one thing you wish more people understood about science or scientists?
Oh, wow. Yeah, that's a great question, especially today when science and academics are under such attack, I mean, so I would say, science is real. We learn stuff. We learn real facts, real information. It's not necessarily the only way to make a decision. But certainly what we do is real and valuable, and I hate to see it being attacked. I teach a class called Science and Ethics in Daily Life, so I pick controversial topics like abortion and AI and use of stem cells and this kind of stuff that, lots of people have opinions on, but probably don't understand the science behind it. And so we talked about the science of it, and I said, you don't have to make your decision based on the science, but you ought to at least understand the science.
Yeah.
And then make your decision based on whatever you think. But don't say the science is wrong, or don't turn the science into something it isn't, because you feel like you have to make a scientific decision. You don't have to make a scientific decision, but you should still understand the science.
Yeah, that's very true. I feel like today a lot of people are very opinionative when they don't even have the full knowledge behind what they're having opinions about.
That's so true.
Exactly, yes. I like to know stuff. I don't like people who tell me you can't know stuff. I'm like, I know stuff.
Well, with that, I want to thank you, Dr. Mulligan, so much for taking the time, even while on this crazy journey you're on right now. It sounds fun. It's literally been on something I've looked at doing in the past myself that I haven't gotten to yet. So I hope you enjoy it greatly.
It's awesome.
And I want to thank our guest hosts, Jailyn and Chloe, for putting together such wonderful questions, finding this great paper. And with that, this has been STEMpaths. I'm Dr. Thornton Larson, and we'll see you guys next time. Thank you.